Bismillahirrahmanirrahim. Today I would like to share the knowledge about plasmid digestion. My name is Sarfrazar Haman. I am a PhD scholar in the Department of Parasitology. I would like to share the knowledge with references these books. Veterinary Parasitology, second edition by G.M. Urquhart. Veterinary Parasitology, fourth edition by M. A. Taylor, George's Parasitology for Veterinarians, 10th edition by D. White, D. Bowman, Cloning and DNA Analysis, 6th edition by T. A. Brown, and last one is the Internet Sources. Please subscribe my channel and also push the button of bell icon so that new video will reach to you. What is the plasmid digestion? Plasmid digestion means that purified plasmid DNA is digested with one or two or more restriction enzymes to give a distinct and clear band pattern. These bands can easily be resolved by electrophoresis. What is double digestion? If two restriction enzymes are used for digestion of plasmid and these show two bands and it can be checked by electrophoresis. What is single digestion? If one restriction enzyme is used for digestion of plasmid and this shows one or two bands depending on the restriction sites, these bands are checked by electrophoresis. What is the restriction enzyme? It is also called restriction endonuclease. It is a protein that recognizes specific short nucleotide sequence and cuts the DNA only at the specific site, which is known as a restriction site or target sequence. These cut within the multiple cloning sites and 2 to 5 bands are selected and these can be checked by electrophoresis. What is blunt end? It is without single stranded tail of plasmid. What is sticky end? It is with single stranded tail of plasmid. What is the plasmid and vector? Plasmid, it is a small extra chromosomal DNA molecule within a cell and it is separated from chromosomal DNA. It has following characteristics. It can replicate independently. It is small circular DNA strand in the cytoplasm of a bacterium or protozoan and sometimes present in eukaryotic organisms. It is used in manipulation of gene in the laboratory, the bacterium having the plasmid carrying genes with genetic advantages to antibiotic resistance, herbicides resistance, metabolism of substrate like beta-galactosidase, etc. It has higher rate of replicating ability. What is vector? It is a DNA molecule used as a vehicle for carrying foreign genetic material into another cell. A vector having foreign DNA is called recombinant DNA. Similarities between plasmid and vector. These are double-stranded DNA molecules. Plasmids and the most vectors are circular DNA molecules. Both plasmids and vectors are self-replicative DNA molecules. Both plasmids and vector can be used to introduce foreign DNA molecule into cells. Difference between vector and plasmid. Plasmid is an extra chromosomal element of mainly bacterial cells. Vector is a vehicle that carries foreign DNA molecule into another cell. Plasmids can also be used as a vectors. Both are self-replicative molecules inside the cell. The gene product of plasmid is not essential for function of bacterial cells. The gene product of vectors is important for the cell. How does the restriction enzyme cut? A restriction enzyme finds its restriction site or target sequence, usually a sequence of 4 to 8 nucleotides. It will stop and leave the DNA molecules, suppose enzyme digestion. The recognition sequence is usually found on both strands, but running in opposite direction, the enzymes will cut both strands. Procedure for double digestion: remove the mentioned or required restriction enzyme, plasmid, and buffer from the freezer and put it into ice until these reach at liquid form. Add 7 microliter plasmid, 1 microliter Zol, 1 microliter Nil, and 1 microliter B4 buffer into 1.5 ml microcentrifuge tube. After mixing, tapping the tube and pulse down in microcentrifuge tube. Incubate it at 37 degrees centigrade for 2 hours into water bath. Load the samples and 1 kb DNA ladder into 0.8% agarose gel for electrophoresis. The following conditions are required during electrophoresis: time 40 minutes, voltage 120 volt, current 400 ampere. After that, visualize the bands at gel illuminator. Single digestion of plasmid with echo or V. Remove the mentioned or remove required restriction enzyme, plasmid, and buffer from freezer and put it into ice until these reach at liquid form. Add 8 microliter plasmid, 1 microliter eco and 1 microliter B4 buffer into 1.5 ml microcentrifuge tube. After mixing, tapping the tube and pulse down in microcentrifuge tube. Incubate it at 37 degrees centigrade for 2 hours into water bath. Load the samples and 1 kb DNA ladder into 0.8% agarose gel for electrophoresis. The following conditions are required during electrophoresis: time 40 minutes, voltage 120 volt, current 400 ampere. After that, visualize the bands at gel illuminator. Single digestion of plasmid with ACC1. Remove the mentioned or required restriction enzyme, plasmid, and buffer from freezer and put it into ice until these reach the liquid form. Add 8 microliter plasmid, 1 microliter ACC1, and 1 microliter B4 buffer into 1.5 ml microcentrifuge tube. After mixing, tapping the tube and pulse down in microcentrifuge tube. Incubate it at 37 degrees centigrade for 2 hours into water bath. Load the samples and 1 kb DNA ladder into 0.8% agarose gel for electrophoresis. The following conditions are required during electrophoresis: time 40 minutes, voltage 120 volt, current 400 ampere. After that, visualize the bands at gel illuminator. 0.8% agarose gel electrophoresis. Take 0.24 gram agarose and mix it into 30 ml TAE buffer. Put the beaker of mixture of solution into hot air oven or microwave for 1 minute. Cover the beaker with papers. Add 3 microliter cyber green dye into mixture. Pour this solution into electrophoresis apparatus. Insert the well comb in to the Agarose solution and wait until it becomes solidified towards the 1 kb DNA ladder and samples into the wells. Run the electrophoresis at the following conditions: time 40 minutes, voltage 120 volt, current 400 ampere. After that, visualize the bands in gel illuminator. Thank you so much.